know because you signed up for the Zoom. Thank you very much for doing so. This is a collaboration between Live with Scientists, which is a non-profit organisation that was
Hi everyone,
at the
thanks
University
for joining.
of Manchester, but we're trying to reach out to as many people
From
as possible.
everybody who's just joined.
And this webinar has been commissioned by
just a couple of minutes
UK-based charity Antibiotic Research UK. So basically, yeah, they would like people to learn about antibiotic resistance, its importance, and one of the charity's main focuses is to try and they want to discover new antibiotics in development. And yeah, so this is a webinar or potentially GCSE levels, but we'll have a few little quizzes and questions, but we're really going to hand over to some experts because I am not the expert. I'll let you know that. So, and just to make a note, it's British Science Week. I'm sure a lot of you are already aware, and it is also our infectious disease month here at Live with Scientists. So that's all tying in nicely. Just to go quickly through the structure of the talk, as I said, we're going to do a quick poll, what do you know already? And then we're going to have a talk from Jess Buddle. So Jess is actually from the University of Sheffield, and she specialises in a bacteria called C. difficile, which I'm sure she's going to tell you a lot more about, as it's a very difficult superbug. After Jess, you'll have a few minutes to ask any questions specifically for Jess and the work that she does. And then we'll do a quiz on Kahoot. Will there actually be a prize for the winner? I'll let you know what the prize is while we're running that. And then we'll go over to Rosie, who is based at the University of Manchester, and she specialises in research in E. coli. We'll then have a Q&A with Rosie and Jess and have a little poll, feedback of what you know, and then, yeah, any feedback from you would be great. So without further ado, I think we'll fill in the poll first. That's what we'll do. So I'm just launching that, and hopefully a poll should have now come up on your screen. And if you could, in your responses to that. So is that up on everybody's screen okay? If you could give me a thumbs up or anything, if it is. Okay, great. People are starting to log that now. And we've got some late comers still coming. It's not too late to put in your poll answers. So anybody who's just arrived, we just did a quick introduction. We've not started the talks yet, but we're just doing a quick poll to see what you already know about antimicrobial resistance. Because Rosie and Jess are going to fill us in a lot more coming up. 26% of you voted. Great. Coming in thick and fast. I hope everybody had a happy International Women's Day yesterday. We've got two fantastic women repping for us right now. Right, I'll just give you another minute to fill out the poll there, please, guys, if you could fill that in. Oh, thank you to everybody replying on the chat as well. I didn't see that. Nearly there. Right, 30 more seconds, and then the poll is going away. So, as I said, we're going to go on to Jess after this, after I've closed this down. And really, I'll just let Jess do the talking because I am not the expert here. It is not my field. But I hope everybody's been having a good Tuesday. I used to host a quiz on YouTube, and I thought I'd be better at small talk. But right, okay. Not everybody's voted. I think there's 10 more people to vote. People take 8 more people to vote. Right. I'm just going to call it a day there, guys, unfortunately, and we'll move on to Jess so we can let her start talking. So I will just stop sharing now, and hopefully Jess will be able to show you her slides. Perfect. I will remove my spotlight and hand over to Jess.
Right, hello everybody. So my name's Jess and I'm going to talk to you guys today about how to kill a difficult superbug. So we're going to start off with a little introduction. I'll kind of tell you a little bit about me and how I got to where I am at the minute. We'll talk about antibiotics, C. diff and resistance. FMT, which if you don't know what this is at the minute, then you definitely will by the end. My own research, and then we'll have a little recap as well at the end. So as I say, my name's Jess and I am a first year PhD student at the University of Sheffield. So I kind of started out my journey at Hempwhite Grammar School. I'm from West Yorkshire, if you can tell. And I did my GCSEs and my A-levels here. And my A-levels were biology, chemistry and psychology. And I also did AS level food and RE, but I know you guys don't really do AS levels anymore. So then I went to the University of Liverpool and I studied microbiology. And this is me when I graduated in 2019. I had a year out in industry, just kind of working as a microbiologist in a factory. And then here I am right now at the University of Sheffield. So as I say, I'm a first year PhD student. And my project is looking at antimicrobial resistance in C. diff, which is a good cue to start talking about antibiotics and C. diff. So I think it's important first to get all on the same page about what are antibiotics and what is resistance. So antibiotics are drugs that are used to kill bacteria or stop them from growing. Either way, you get the skin, the person's cured, they don't have the disease anymore. They work by targeting structures that are specific to bacteria. So they won't do anything towards human cells, they can't harm us, and they don't do anything whatsoever towards viruses. So if you went to the doctors with a common cold, then you wouldn't get antibiotics because it literally wouldn't do anything. So, yeah. So there's two different types of kind of categories of antibiotics, broad spectrum and narrow spectrum. Broad spectrum target a structure on the bacteria that is common to most bacterial species and they will kill many different types of bacteria. Narrow spectrum only targets something that is specific to one or a few species of bacteria, have different kind of benefits and downfalls, I suppose. So resistance occurs when bacteria evolve methods and means of protecting themselves from being killed by antibiotics. So this is kind of a natural phenomenon, evolution. You know, if bacteria in the environment and there's antibiotics present and bacteria can survive these antibiotics, then these ones will be selected and so the process goes on. However, the misuse of antibiotics by humans for both our own use and for animal use in, you know, farm animals, zoo animals, pets and everything, this misuse has rapidly accelerated antibiotic resistance. And it's actually now one of the major threats to global health today. So it's a really big problem. So what is C. diff then? So C. diff, I probably should point out that this isn't really the exact order I'll be talking in, but it just kind of look cool. So C. diff is a bacteria and it's an anaerobic bacteria. And this basically just means that it can't survive in the oxygenic atmosphere that we're in right now. So you won't find C. diff live on surfaces. It's solely, well, anaerobic, so it can survive in the gut and things. So what does this then mean? Well, for me as a scientist, it means that I can't study C. diff on the bench like you would with other bacteria. I have to study C. diff in an anaerobic cabinet, which is just essentially a big plastic box that's see-through and you put your arms in, it's got no oxygen, and you can study C. diff like that. For C. diff though, what does this mean? Well, if it can't survive in the atmosphere that we're in right now, then how is it possibly transmitted between people? It's actually transmitted by spores. So these things here are spores, and they're just basically little packages of bacteria, little packages that will eventually germinate into a bacteria. And these are really well evolved to be transmitted. They're pretty much resistant to anything, like hand sanitizer, oxygen, heat. They're really, really well evolved to be transmitted. C. diff is also a really difficult bacteria to treat, and this is for a number of reasons. So it's partly because it's resistant to different types of antibiotics, but it's also difficult to treat because the disease is often recurrent. And this basically means if someone is cured, then the disease can come back again and again and again. And the more times it comes back, the worse the outcome is for the person. So it's difficult because the more times it comes back, it's harder to treat, and also the person is more likely to die. So after about 3 reoccurrences, the person has a 50% chance of survival, which obviously is not very good. So why is C. diff cool then, but not if you have it? So C. diff causes a complete range of symptoms, literally from absolutely nothing to death. So it's like the complete range. Most people who have C. diff will get diarrhea. If it gets really bad though, you get something called pseudomembranous colitis. And this is what this picture here is showing. So this is actually a picture of the inside of someone's gut. Now, if you imagine your gut and my gut, it's nice and pink and it's smooth and healthy and it's absolutely not covered in these big yellow blobby splodge things. This is just not good. It's not good for the person. It's not good for like the gut. It's not good just in general. You would not want this disease at all. And if this progresses further, then it ends up in death. So this generally affects people that are older, that are already ill or in hospital. However, community-acquired C. diff is becoming more common. And this is C. diff that isn't necessarily associated with hospital, and it's affecting people that are younger and also healthier. So let's talk a bit of numbers then. So C. diff causes around 453,000 cases a year, 29,000 deaths a year, and it costs up to $3 billion per year in the USA alone. So this is just one country and 1 year, and it costs $3 billion. And this is only the direct costs. If you add in all the indirect costs as well, then it ends up being $6.5 billion a year. So this is obviously a lot of money and a lot of deaths attributed to just one single bacterial disease. And we obviously want to reduce this. So let's talk about resistance in C. diff then. So C. diff is resistant to a lot of different antibiotics, things like cephalosporins, clindamycin, fluoroquinolones. Not that you really need to know what these are, but these are different antibiotic groups that C. diff is resistant to. And what does this mean? Well, it means that patients with C. diff infection can no longer be treated with these antibiotics. So this reduces the treatment options available. And it means that if C. diff ends up being resistant to all antibiotic groups, then we'll no longer be able to treat the disease. So if someone gets C. diff, they'll end up either dying or surviving, and there'll be absolutely nothing we can do about it. It'll literally just be like going back to an era before antibiotics even existed. And we really don't want this to happen, because this would be really, really bad. And the deaths would, it would be terrible. You might also notice at the bottom here, metronidazole is in a question mark. And this is because this drug is actually still used to treat C. diff in mild cases. If you got a C. diff from a patient in hospital, it's generally, it's generally not resistant to metronidazole. However, resistance is on the rise. So this is for a couple of reasons, partly because of like mutations which make it resistant, but it's mainly because of a plasmid called PCD-METRA. So a plasmid is just a circular piece of DNA. It normally contains genes for resistance, and it can be passed between bacteria. So this plasmid PCD-METRA, responsible for metronidazole resistance, is being passed throughout C. diff, and it means that resistance is increasing. So if resistance becomes widespread, then this will be another drug that we can't use anymore to treat C. diff. So this is a really, really, really big concern, in case you haven't gathered that from how many times I've said it. However, I've kind of painted this nice image where someone has C. diff and they have antibiotics and they get cured, and that's fantastic. And in reality, it's not exactly that simple. So C. diff and antibiotics have a bit of a strange relationship. And this is because antibiotics are both the cause and the treatment of C. diff infection. So how can this be? How can antibiotics cause C. diff but also treat it? Well, if we start at the top here, someone has a disease, and it's not C. diff, it's just random disease of the gut. And this person gets antibiotics, and this is great because it means that this disease goes away, person's cured. Well, that's great, right? However, you guys might be aware that humans are just a giant bag of bacteria. We're more bacteria than human. We've got loads of bacteria, and most of these are really, really good for us. We wouldn't survive very well without them. They do lots of things for our digestion, for vitamin production, loads and loads of good things. So when someone gets antibiotic treatment, not only does it kill this bacteria, but it also kills loads and loads of and this obviously is really bad. It means there's no nice bacteria to protect against C. diff. So if you imagine your gut as just a giant room full of people, and there's a person in the room because the room is just jam-packed full of people. However, if antibiotics come along and basically everyone in the room leaves, then the room's now empty, and the person called C. diff can enter the room and do whatever they like. And this is essentially exactly what happens in the gut. So if there's no nice bacteria to protect against C. diff comes in, causes infection, and then antibiotics treat C. diff. As I say, it's a bit of a strange relationship, and there isn't very much we can really do about this. And when I say not very much, there is actually one alternative to antibiotics, which is fecal microbiota transplant, FMT. This is exactly as it sounds. It's literally taking the feces of someone that's healthy and putting it into someone with C. diff in the hope that the bacteria from the healthy person's feces will help to kind of fight off the C. diff infection. Now, obviously I'm presenting and I can't really see your guys' faces, but I assume you don't really like the idea of swallowing someone else's poo, and obviously you wouldn't be alone. So the public acceptance of this treatment is not very high. There's also a few worries. Well enough, then you might be putting more diseases into someone who's already ill, which obviously isn't ideal. So yeah, it is a cool treatment and it is viable, and they're trying to, get it off the market a bit, but this is not commonplace yet. And this is pretty much the only alternative that's really, viable for C. diff treatment. So the bottom line is that antimicrobial resistance is a really serious issue, and we need more antibiotics, and we need them as soon as possible. So let's talk a bit about my research then. So I work on 2 antibiotics that are used to treat C. diff, vancomycin and RDZ. Vancomycin is the current frontline drug to treat C. diff. So pretty much most cases get treated with vancomycin. Recurrent cases, severe cases, vancomycin is the frontline drug. The other antibiotic is RDZ. So this is actually a novel antibiotic that isn't on the market yet. It's in phase 3 human trials, which means that hopefully it'll go on the market in the next couple of years. So what do we want to know then? So we want to know what these antibiotics target in C. diff. We want to know how C. diff how these antibiotics kill C. diff, which is the mode of action. And we also want to know the fitness costs to developing resistance. So C. diff might be able to develop resistance just fine, but this might mean it has a fitness cost that makes it less viable to kind of surviving in the environment. And this might be why we haven't really seen vancomycin resistance yet, for example. So these are really big questions, and we really need to know a lot of the answers to this to try and stop antibiotic resistance. So how do we do this then? So this basically involves a laboratory evolution experiment where we evolve resistance to the antibiotic in question. This involves a lot of plates like this. You put in the media, the bacteria, and the antibiotic into a plate, and the bacteria that are resistant will survive. And you put these onto a new plate with higher antibiotic, and then just repeat the process basically again and again until eventually you have an evolved population of bacteria that are resistant to the antibiotic in question. So when you have your evolved population that are resistant, you sequence all of the DNA in the evolved population and all of the DNA in the original bacteria. And once you've got both of these, then it's essentially just a glorified, very complicated game of spot the difference. So please don't actually do this, just disclaimer. It was just proving a point that basically you're looking at the DNA of the evolved and the original bacteria and spotting differences between them. So what does this really tell us? For example, if you have a gene that has a lot of different mutations in it, then this might mean that this is the antibiotic target, and in mutating the gene a lot of times, C. diff has been able to become resistant. So you kind of work backwards like that and then do a lot of downstream experiments. And as I say, this can tell you the target and the mode of action of the 2 antibiotics. So what else can you really do with this then? So eventually we can use this to predict when resistance is about to arise. And you can do this by looking at DNA diff in hospital, so the C. diff DNA from patients, and comparing it to all of the data that I've found. And then if there's mutations that are common, then we might be able to say, well, resistance is going to arise very soon, so that then we can inform the treatment choice, swap to a new antibiotic, and hopefully this will reduce the spread of resistance and basically prolong the antibiotic lifespan. So I'm kind of nearing the end now then, so I'll just give you a little recap. So C. diff is a very important bacteria, and it causes lots of different things but mostly diarrhea, which can lead to death. Antibiotics have a bit of a strange relationship with C. diff. They're both the cause and the treatment. And C. diff is resistant to a lot of different antibiotics, and this makes finding new ones very, very important. And we can use experimental evolution in the lab to help find both the targets and mode of action of antibiotics, and eventually this will help prolong the antibiotic lifespan. So that's it from me. Thank you all for listening, and obviously thanks to everyone here, and I'm happy to take any questions. I'll stop sharing now so I can see you all, hopefully.
That's great, Jess. Thank you very much for that. So yeah, if anybody's got any questions for Jess or any general C. diff questions, or you've I don't know, anything confused you there, feel free to type them in the chat. One person has asked, could C. diff be treated with bacteriophages?
Oh, this is a brilliant question. So I don't work very much on phages. I have a few people in the lab that do that, and it's not really my area of expertise. But phages are, well, they're really a kind of promising alternative to antibiotics. There's not been a lot of success as far as I know just yet, but watch this space, because in the next however many years this might be the only option we have if we can't find new antibiotics fast enough. So yeah, really good question.
Great. I'll just take this opportunity to introduce Alex as well. So Alex is going to help take some questions with me. So Alex, can you see the questions in the chat? Do you want to read out the next one?
Yeah, I can do that. So I got a question from Donna, who is asking, if C. difficile is an anaerobe, how is it contracted through food surfaces, etc., where I guess they're exposed to oxygen?
Yeah, great question. So basically, C. diff are transmitted by spores, and spores are just like little packages of stuff. And basically, these spores are resistant to oxygen. So when the person ingests the spores, they go all the way through to the gut, and then they can germinate into a live bacteria and then cause disease.
Nice one. Should we keep going with the questions, Casey?
Yeah, if we keep going with the questions, I'll just share my screen because we're going to do a Kahoot quiz in a moment. So if people want to start logging in while Jess is answering some questions, hopefully you can all see that there. Let me know if you can't, but yeah, if people want to start logging into the quiz, that would be great.
So we've got a few questions, Jess, about probiotics. So Ayana and Sonia have both asked if, Ayana is asking if probiotics can be used to treat C. difficile, and Sonia is asking if prebiotics can help fighting C. difficile infections.
These are, I'm very impressed at how much you guys know. So these are really good points. So things like probiotics, prebiotics, etc., potentially could be viable in the fact that the whole reason people get C. diff is because they don't have a lot of bacteria in the gut to protect against it. So potentially viable. However, we don't really know enough about the microbiome, I would argue, that we can bacteria that won't harm too much else in your gut. So there is actually a paper that kind of published different views on this. So basically looking at probiotics, looking at no treatment, and then looking at fecal microbiota transplant. And it actually found that probiotics are a bit detrimental to recovery of the microbiome instead of so fecal microbiota transplant was really good. Probiotics is actually worse than just letting it recover naturally. So there's a bit of kind of confusion in this, and I don't think we have enough knowledge really to put this forward just yet.
Very interesting, yeah. So Harrison is wondering if there are any precautions being put in place to reduce the spread of spores.
So there is a lot. So C. diff is kind of a bit like a lot of other diseases in the fact that you get epidemics where you get sudden spikes of C. diff disease. And there is a lot of money spent on sanitizing everything and being able to properly kill all these spores. So it's a really good question. And there is a lot of money spent on trying to reduce the spread of it. If you've ever been to hospital as well, you often get a little swab before you go into hospital, if you're going for an operation. And this is to test for MRSA and C. diff to make sure that we're not bringing it into hospitals as well.
Excellent. So a question here from Catherine, going back to the phages. Catherine asks, do you think bacteriophages will become a treatment in NHS in the UK? How long do you think it will take for phage treatment to become available through the NHS?
Good question. Hopefully there's a lot of work going into phages being used for an alternative to antibiotics. New antibiotics are very few and far between. So if we can actually get this off the ground, then it'll be really, really helpful to reduce, obviously, the bacteria that can't be treated anymore. So potentially, yes, and hopefully within maybe about 10 years, but these things do take a lot of time. So I couldn't really put a timestamp on that for you.
Yeah, it's hard to predict, isn't it?
Yeah.
Ria is wondering, how dangerous is it evolving C. difficile in the lab?
This is a very good question. So there's different types of C. diff. Some of them are not very pathogenic. Some of them are really pathogenic. The ones that are pathogenic, sort of the epidemic strains that spread really well, obviously are more dangerous, but these are more clinically relevant. So when I'm evolving my C. diff, I'll be using clinically relevant strains, and it could be really dangerous. You could cause a new pandemic of really resistant C. diff, which we don't want. So all of my work that I've been doing so far is actually just to knock out a few different genes to make sure that it doesn't cause disease. So even if it did possibly get out, then it wouldn't do anything. Great question. Yeah, thinking of all the things that I had to think about before I started this project, yeah.
Sounds like you're being very responsible. Let's have a look here. So Zongo is asking, for the experimental evolution, do you use a constant dose of antibiotic? Do you cycle which antibiotics they're exposed to? And do you do any work where you don't treat them with antibiotics?
This is, wow, very advanced. So I've read a few papers that do cycling. I haven't actually started this yet. As I say, I'm still kind of a non-pathogenic C. diff that we can use to evolve, which takes a lot more work than you'd probably think, with it being an anaerobe and hard to study and such like. But yeah, so my plan at the minute is to do constant dose and also evolve in parallel with no antibiotic, because then you'll be able to see what changes are due to the antibiotic and what changes are just due to the environment, or, just random mutations. And that'll help make sure we follow the interesting things rather than the, rubbish things.
Definitely. We've got so many questions coming in.
Yeah, I was going to say, if we just maybe do one more and then any more we can maybe pick up. I don't know how many might be still relevant to Rosie as well.
So final question, I guess, before we go on to the quiz then. Geneva asks, are there any other ways to extract and to transplant bacteria from a healthy person to treat C. diff that isn't a fecal transplant, something that's maybe more palatable?
So there is a few different kind of variations on fecal microbial transplant. You can put it in as an enema. You can put it in, dry it up and put it in a pill. I mean, none of these sound particularly amazing, really, do they? But there is kind of like a lot of research going into how to make it a bit more, nice. And there is a lot of research as well into obviously the gut microbiome and trying to make kind of a cocktail that would be prebiotics, but also a really good range of them that kind of mimics the microbiome, all your bacteria in your gut, which might be obviously a bit nicer and people might accept it a little bit more. So there's a lot of work going into it.
Excellent. Okay, so I think I can see the details for the quiz, so I imagine everyone else can as well.
Yeah, you can see the slide, can you? Yeah, so I can see that one or two people creeping in at the last minute here, but yeah, we'll start. Loads of people are just having a flurry of joins now. Okay, we've got. I'll give you one more minute, everybody, to join. But yeah, thanks very much for that, Jess. That was really, really interesting. You hear a lot about C. diff, but I don't actually know too much about it. Okay, so I think the numbers have paused there. So I'm going to start the game. So hopefully everybody joined in who wanted to join in, and you should be able to see the questions come up on your screen. Question, if people can't see it on the screen, is: do antibiotics only kill bad bacteria? True or false? Hopefully Jess just give you a good lay down on that. Okay, most people got that one right, which is fantastic news. Question number 2. So you might not know this yet because Rosie's not talked yet, but what does the E in E. coli stand for? Got 4 options. Is it everywhere? Is it endospore? Is it endoscopy? Or is it Escherichia? Should have practiced that one. I'm sure Rosie will.
You nailed it.
Definitely did not nail that. Right, okay, we've got some mixed responses there, so I'm sure Rosie's going to put us all right in a minute. A reminder, there is a prize for the winner of this one as well. So you should all know the answer to this one because Jess just told us, but how is C. difficile transmitted? Is it fake news? Is it animals? Is it spores or is it meteorites? So hopefully we get a good rate of right answers here. Yes, pretty much everybody got that right. Very well done. So a reminder, the quicker you answer, the more points you get in this quiz as well. Should have probably said that at the beginning. What does FMT stand for? All right, we're all answering this one pretty rapid. I think this one's stuck in people's minds. The options are fight microbial transmission, fecal microbiota transplant, failed microbe testing, or face-mouth transmission. It was, of course, the fecal option. Got some good scores going on. I think you should be able to see on your screen how well you're doing if you're playing along. This question is, which of these is not a bacteria? So is it C. diff? Is it tuberculosis, E. coli, or polio? What could it be? Most people are answering pretty quickly. I think we've got 100 people playing along. It was polio, quite a big split there. I think some people didn't read the question right because people have put C. diff as an answer. Oh, person in first place has just switched around. Who could it be in the end? Which of these is not an antibiotic? Oh, sorry, which of these is an antibiotic? Don't listen to me. Is it vancomycin, paracetamol, antihistamine, or hermahedicin? Okay, a couple of seconds left on that one. It was, of course, vancomycin because Jess was just telling us about that one. Oh, it's tight up at the top, guys. 2 more questions to go. So question number 7, bacteria can exchange DNA with other bacteria, true or false? 10 more seconds on this one. Oh, I think everybody's answered anyway. True or false? Guess if you don't know. It was, of course, true. Just swapping them genes around like it's nobody's business. And final question. In the last 40 years, only 2 new types of antibiotics have been discovered. Is that true or false? Everybody's answering so quickly. Right, fantastic. So, whoever the winner is, please, can you privately message me on the chat? If you're watching via Zoom. If you're not watching via Zoom and you're on YouTube, you can email us and I'll get that up on screen now. So first place, you know who you are. It should have popped up on your screen. And there is a Gmail account up there that you can see. And if you're on YouTube, can you please contact us via that, what your nickname was, and then we can verify that you're a winner. And I will let you know what you've won, and everyone will be very jealous, I'm sure. Okay, so like myself, if you need that email address, it will come up at the end again. But Alex, do you want to introduce Rosie? Kind of did a little lowdown before.
Yeah, absolutely. So Rosie is a second-year PhD student at the University of Manchester. So she works with Dr. Chris Knight and Dr. Guilford, and she is researching antibiotic resistance using E. coli. So you want to take it away, Rosie?
Yeah, I've just got to get this to work. Oh, can you all see that?
Yep, that looks great.
Now, got to learn how to full screen. That could be a problem. Didn't actually check that. Oh, there we go.
Nice one.
So I'm Rosie, and I'm going to give a talk on the cost of antibiotic resistance. So that's going to be like a whistle-stop tour of just kind of all the research that I've done in the first year of my PhD. But I'm going to start off with a bit about me. Yeah, a bit about me. So I have a bachelor's degree in biomedical science and went to do my master's degree in medical microbiology, where I learned to love microbiology, specifically E. coli, as you will see going on. And now I'm a second year PhD student in ecology and evolution at the University of Manchester. This is antibiotic resistance evolution, but particularly multi-drug resistance in a clinical set of E. coli. So those are E. coli that have come from patients, which is nice. So a bit about what I'm going to be talking about. So I'm going to talk about E. coli and multi-drug resistance. So you're going to understand all about multi-drug resistance by the end of it. And then I'm going to go over what the cost of resistance is, because it's not a term that's usually spoken about normally. When you think of cost of resistance, usually people think of like actual cost, like physical money. And that is not the case in this talk. And then I'm going to talk about how I test for a resistance, how antibiotics affect the growth of bacteria, and what I found out. So let's move on to E. coli, or Escherichia coli. Good job, Katie, trying to pronounce it. So they're a gram-negative rod-shaped bacteria, and they normally live in the intestines. They're a part of a family called the Enterobacteriaceae, if I can say that right. And as everyone probably knows, they commonly cause stomach bugs. But I'm interested in the urinary tract infection cause. They cause about 90% of urinary tract infections. And if a urinary tract infection is severe enough and it travels up further, it can cause pyelonephritis, which can lead to bloodstream infections. And about 70% of bloodstream infections are caused by E. coli. So how do we treat E. coli infections? Now, this looks like a very scary piece.
I think your slides might have frozen.
Oh, no.
Yeah, we're just seeing the first slide.
Oh, absolute nightmare. Give me a second.
It did make sense.
Let me fix it.
It's other people pointing it out on the chat.
Right, let's try this again. Can you see
everyone on the chat? We can see the first page, yeah.
Okay. Oh, it's not going to... Oh, technical difficulties, folks.
Don't worry about it.
This is why I'm not good at giving presentations. All the technology, isn't it? Can you still see the first? It's just not letting me present now. Oh,
you can flick through them like this.
Oh, wait, worked out the problem. It's Linux for you. Doesn't like working. Can you see it now?
I think it might still be the same as it was before. We can see the first one.
If I'm moving through, can you see me moving through?
No.
Oh my God.
If you just look through the slides.
Right, is it stopped screen sharing?
Yes.
Okay, right, let's fix this. Right, if I try this one. Right. Can you see this screen?
We can see a different slide now.
Right, can you see this one?
Mm-hmm.
Can you see it full screen now?
Yes.
If I flick through, can you see it?
Yes.
Yay!
Woo! So where did it stop at? And then we'll start.
We never got past the first slide, I think.
Right. So you didn't see this one?
No.
Absolute nightmare. Yeah, so here's everything I'm going to talk about. I might as well just go over it again quickly so you can actually see what I'm talking about. So yeah, E. coli multi-drug resistance, cost of resistance, how we test for resistance, antibiotics and how they grow, and what did I find out? Now, E. coli is a gram-negative rod-shaped bacteria, as I've just said, and it normally lives in the intestines. It's a member of the Enterobacteriaceae, causes food poisoning, but I'm interested in urinary tract infections, and they cause 9% of urinary tract infections. So yeah, moving on from that, how do we treat UTIs? And yeah, this looks very scary. It looks like a scary piece of writing, but they are the greatest things to look at. So the National Institute of Health and Care Excellence, or NICE if you like your acronyms, produce, for every infection that you know, they produce a set of antibiotic prescribing guidelines. And that'll tell you what the first line antibiotic is that you should be using, the second line antibiotic you should be using, and all these antibiotics for all different ages. And it's, honest to God, it's the best piece of information. So we can tell, for instance, nitrofurantoin is what is used, and trimethoprim. And for pyelonephritis, which is the more advanced UTI, which can lead to bloodstream infections, cephalexin, coamoxiclav, trimethoprim are usually used as first-line antibiotics in this case. So moving on from that, we're going to discuss antibiotic resistance because it seems like it's a nice follow-on. So to progress from what Jess was saying, antibiotic resistance describes a change in bacteria that makes them makes antibiotics a little less effective. So in 2020, they recorded 700,000 deaths as a result of antibiotic resistance globally. And there's a prediction that by 2050, that'll increase to 10 million. And that will cost, across Europe, North America, and Australia, £2.7 billion, which is a lot of money. And that is why research like this is so important. So antibiotic resistance in hospitals. Now, this is important to me because the E. coli that I use come from clinical organisms. So antibiotic resistance is a really significant issue in hospitals. And the reason for that is that there's a lot of people, there's visitors, there's healthcare staff, there's a lot of movement around a hospital, which can bring in organisms. There's a lot of very, very, very sick people. And that obviously being immunocompromised means that they're more susceptible to those infections, especially being elderly. A lot of elderly people end up with UTIs and they're hard to treat. And there's a lot of heavy antibiotic use in hospitals, obviously, because it's a hospital. And that makes bacterial infections harder to treat. And that means that simple infections like a UTI or say a surgical site infection, if caused by an antibiotic resistant bacteria, eventually it's going to be super, super hard to treat. And then we get to multi-drug resistance, which I'll get onto in a second. And then it's going to be impossible to treat. And then you get people dying from very, very simple infections, which is not what we want. So multi-drug resistance. Multi-drug resistance is similar to antibiotic resistance, only it's the ability of bacteria to counteract the effects of multiple antibiotics, multiple key antibiotics that are used to treat them. So in my case, oh no, we're going on to the cost of resistance now. I don't know my slides very well. The cost of resistance is a physical impact of antibiotic resistance. So we can physically see this, and it impacts the ability of a bacteria to survive in the absence of antibiotics. And it can affect multiple biological functions in a bacteria. So you've got reduced competitive ability, which when you think about this experiment mentally, which is what I do, competitive ability, when you've got two strains of E. coli and you put them together and you measure whether or not they can compete, it's very hard to tell which one's which. And then you've got reduced virulence. And that's, when we talk about virulence, we're talking about a bacteria's ability to cause damage to a host. And yeah, that would not be very ethical to test because then you'd have to infect a load of humans with these bacteria, and that's not great. So the one that we go for is impaired growth. We want to know how gaining resistance to antibiotics impacts a bacteria's ability to grow when you take away those antibiotics. So that is what I mean when I talk about the cost of resistance in this. And a bit about antibiotic stewardship, because this is where it all stems from. When we talk about antibiotic stewardship, I'm pretty sure everyone's seen the Keep Antibiotics Working stewardship programme that goes around with the crazy advert with the dancing pills. So yeah, that is one of the campaigns to try and reduce antibiotic use in the community by the government, Public Health England, WHO, all over the world, about different ways in which stewardship is input. But they all come down to three major points: access, watch, and reserve. So you've got access, and that's providing access to first and second line antibiotics initially. Second is watch, and watch means that you keep an eye on those antibiotics that have a higher potential for resistance. Some antibiotics in some organisms are more likely to become resistant to some of the key antibiotics. So it's keeping an eye on those and knowing which ones to look for. So before you prescribe an antibiotic, you're testing to see whether or not that organism's resistant to it before you even prescribe it, which would seem like common sense, but has only recently been adopted. And reserve, which means reserving those last line antibiotics, those ones that have no resistances, no resistance mechanisms known, saving those till absolutely essential, till no other antibiotics work. And our current studies have shown that reducing antibiotic therapies in response to rising resistance rates reduces the spread and the spread and dissemination of antibiotic resistant organisms through clinical environments, so through hospitals and such. And so the purpose of my research is to answer the question of how does multi-drug resistance affect bacterial growth when you take away those antibiotics? If you take antibiotics out of those systems, how does the bacteria behave? So the organism that I've been working with is 91 strains of E. coli, which we collected from Wythenshawe Hospital in Greater Manchester. We didn't know exactly where these came from, but we assumed that they came from either a bloodstream infection or a UTI, because when you consider E. coli, you don't really go to hospital for a stomach bug. So it's the organisms that we're working with are going to be taken from hospital if we were working with stomach bugs. And the antibiotics that I've decided to work with are nitrofurantoin, because that is a first-line antibiotic, along with trimethoprim, which is a first-line antibiotic. And then we looked at second-line antibiotics. So we've got fosfomycin, ciprofloxacin, and colistin, which is the last-line antibiotics, not a very nice antibiotic. And then we looked at ones that are commonly used to identify resistance mechanisms. So cefpodoxime is in the same class as penicillin. And that tests for something called extended spectrum beta-lactamase producers. And they're a class of antibiotics that are able to produce an enzyme that eat all of the antibiotic away, basically. And so that tests for that, which is actually quite a common mechanism of resistance. Meropenem, which is also in the same class as the extended spectrum beta-lactamase producers, but this is the last line of that class. So it's not a common resistance that you come across. And when you do, they're known as CPE, or Bacteriaceae, which is quite a cool acronym. That's what I meant to say. Gentamicin, and then we went for nalidixic acid and rifampicin, because those are what are used to test mutation rates in our lab for a comparative eventually when we get there. And we expected to see one of two outcomes from our research. The first outcome would be that we saw a decrease in growth when you increase the number of resistances in an organism. The second one would be that acquiring that first single resistance causes such an impact to that bacteria that it affects the growth so significantly that even if it got 10, 12 resistances, it wouldn't make a difference to how it grows. I hope that made sense. Right, so how did we test these resistances? And this is where it gets to the really interesting, juicy bits. So again, another table that looks really scary, but I will explain it and it'll all make sense. So there's a standard called UCAST, and it stands for the European Committee of Antimicrobial Susceptibility Testing, bit of a mouthful. And they produce guidelines on every single antibiotic that you can think of. And it'll tell you at what concentration of antibiotic that strain will show up as either sensitive or resistant. And so I use this table to come up with a single value of antibiotic in which I can pop into agar or into a broth and then grow up my bacteria. And if it grows, it's resistant. And if it doesn't grow, then if it grows, it's resistant. If it doesn't grow sensitive, my brain went to mush then. And it'll here, I hope you can see my cursor. It'll tell you exactly how to grow it, which is perfect. That's exactly what you want. And if you ever want to know how to grow bacteria, to know whether or not something's resistant, these are the place to go. So we used, for our testing, we used Muller Hinton, which is a standard for antibiotic testing. And the reason that that's the standard for antibiotic testing is because the ions in it. So when you think about magnesium, calcium, sodium, like the makeup of a broth, they're all really, really tightly controlled, so they can't affect how an antibiotic works, which is really good. And it also contains starch, and starch absorbs the toxins that are produced by a bacteria. So then those toxins can't then counteract the antibiotic as well. So you are getting a true reading of whether or not something is resistant or sensitive without interactions from bacteria or the broth itself playing a part. And again, we used antibiotics that are commonly used to treat UTIs or testing for common resistance mechanisms. And some cool photos now. So this is what it would look like if you put no antibiotics into an agar and grew up. So each dot is a single strain of E. coli. So there are 91 strains on this lovely little piece of agar. And yeah, so that's how they grow if you don't put any antibiotics in. Now, if we move on to the next slide and everything loads nicely, you can see how they grow when you put different antibiotics in. So underneath, it'll tell you exactly what antibiotic it is. And you can see exactly how they've grown. So those are the same 91 strains in different antibiotics. So you can see for nitrofurantoin, there's no growth at all. And that correlates with the fact that it's a first-line antibiotic. So we want that. We want a first-line antibiotic where there's no resistance. The other first-line antibiotic, which is the questionable one, is trimethoprim. So there are really, really, really high rates of resistance within E. coli, and it shouldn't really be used as a first-line antibiotic when you're treating a UTI infection. Although when you look at the guidelines, and when you look at regional guidelines as well, so they also have antibiotic regional guidelines. So you can go on to any district, so Greater Manchester, for example, and you can see how they prescribe antibiotics for different infections as well. It's really, really interesting. But yeah, they also prescribe trimethoprim as a first-line antibiotic with the guidance that resistance should always be tested beforehand. That's the most important thing, is that especially in antibiotics like trimethoprim or anything that's like penicillin in the same group as penicillin, because resistance rates are so high, it's really important that you test for resistance before you prescribe. But obviously nitrofurantoin you go to. And you can see here that colistin doesn't really have any growth, but colistin's quite a nasty antibiotic. You wouldn't really want to prescribe that as a first line antibiotic. So yeah, then we went on to do the same thing but in broth. And the reason we did this is because it acted as a direct comparative. So we could take the numbers of resistances that we saw for each of the strains on the agar, and we could compare that to how they grew when we take all the antibiotics away in broth. So we grew them in antibiotic-free growth for 24 hours, and that produced an area under the curve. And now the area under here, and it's just the space underneath the red line of each of the organisms, and that produces on a graph. And also this bottom one's really cool because that's how they grow. So you put them all in a 96 well plate, and that's how they grow after 24 hours. And you can see that all of the cloudy ones are where they've grown, and then you've got a few blanks shoved in for extra measure. So then we compared the number of resistances that we got on agar with the growth that we saw in the broth. And what we expected to see was a decreased growth as the number of resistances increased. If you can remember back to the slide where I talked about the hypothesis and what we expected to see, this was a negative trend. So we should have seen a slope that went like this, woo, down here. And we did not see that, which is fabulous, isn't it? So what this graph is telling you is that as the number of resistances increase, the growth remains the same. I know it looks like it's slightly going negative, but the slope varies around 0. So that's showing us that the number of resistances doesn't affect growth. Fabulous, isn't it? Then we go on to a bit of phylogenetics, and this is where things start to get slightly complicated. So I'm going to try and explain this the best that I can in the simplest way I can. So phylogenetics requires whole genome sequencing, and it's the study of evolutionary relationships. So how related are two strains of bacteria to each other? And I'm pretty sure everyone's come across the tree of life before, but that's how we're all related to everything else that lives and such. But you can do that with a single set of bacteria. You can work out how related they are to each other. And so what did we want to ask by doing this? Because it makes no sense just to do it. We've got to have some questions, haven't we? So we want to know, do related bacteria share the same number of resistances? Do related bacteria grow in a similar way? 2 simple questions to answer. And you can do that by plotting the number of resistances to where things sit compared to where they're related, if that makes sense. So this is the first one, and it looks really confusing, but it's not. It's a phylogenetic tree. It's a bit like the tree of life. So here you can see that all of them cluster together, and where they cluster shows how related they are to each other. So what we'd expect to see is, for example, take this cluster here. They're all related to each other, and if resistance followed relatedness, so bacteria that are more related to each other have the same number of resistances, we'd expect everything on this little cluster here to have the same number of resistances. But that's not the case. And now if we look at how growth compares, this is a bit smaller because it's quite a large one. Again, looking at the same cluster, you can see slightly different colours, which means that, again, relatedness doesn't play a role in how things grow, anything else playing a part. So I'm coming to the end of my talk now. I'm just going to go over what we found and why it was important that we found it. So we found an apparent lack of fitness cost or cost of resistance associated with the number of resistances. So what does that mean for resistance? So if we think back to antibiotic stewardship and this whole halting antibiotic therapies will stop the spread of antibiotic resistant bacteria, that might not be as simple as what we're saying. There must be something else that's causing this lack of negative association, if that makes sense. And then we saw no significant clusters of resistance in related strains. So we're not seeing any clusters here of the same colour. And that could be because of this whole idea that bacteria can share genetic information. So just because 2 bacteria are related to each other doesn't mean that they haven't gained information from other bacteria that they've been in close contact with. So that shows us that mechanisms such as horizontal gene transfer, which is the transfer of genetic information from one bacteria to another, are playing a role in resistance. And the strains all appear to grow independent of relatedness. Now, what's causing this? And again, it's all to do with the type of genes and the association of genes to what we see experimentally that plays a role in it. It's a lot bigger than just saying that if we stop antibiotics, it's gonna change everything. It's gonna sort everything out. It's a lot more than that. Of what I don't actually know at this point, because there's a lot more that's got to go into it. And yeah, that's pretty much it.
Well, brilliant. Thank you, Rosie.
I hope that made sense.
Yeah, no, that was great. So there's already been a few questions for Rosie that have come through. So maybe we'll start with a few questions for Rosie and then open it up to questions for both of our speakers. So let me have a look here. So Kim's got an interesting question. So how much of a role does the livestock industry play for creating antibiotic-resistant bacteria?
Well, that's a really difficult one for me, because my focus is on clinical bacteria. So in terms of E. coli that has become resistant in, say, an agricultural environment, that's not something that I've particularly looked into. I'd say it plays a significant role, but when you think about like clinically, it may be a lesser of a role.
That makes sense. Thanks for that. So we got a question from Scott who says...
I'm just going to put up the poll because I'm just conscious that we're running over time. So I'll just put up the end quiz if people want to do that while they're listening to Rosie's answers.
That sounds great. So Scott is saying that there's some evidence of resistance to trimethoprim slowing down due to reduced use. Do you think, Rosie, that resistance could be reversed even if we continue to reduce our use of antibiotics?
I don't think resistance can be reversed. No, I think it's there are a lot of factors that can play a role in a bacteria being able to lose resistance. That's possible because of the way that resistance works, and so resistance can be held on something called a plasmid, and a plasmid is immobilizable. It can move basically. It's a movable piece of DNA. And sometimes bacteria can lose that through generations of growth. They can lose that thing that makes them resistant. So that's a possibility. But I think to say that antibiotic, it might be to do with the fact that if you take the trimethoprim out of the system, then those bacteria that are resistant to trimethoprim just can't grow as well. It's a little less to do with them not being there and a little more to do with them not being able to grow as well.
Excellent. So we've got a question from Eleanor. This might be something that Jess can weigh in on as well. But Eleanor is asking if T4 bacteriophages could be used in the future if E. coli develop severe levels of resistance. I don't know what a T4 bacteriophage is, but hopefully that makes more sense to you.
Bacteriophages are something that I've never really worked with. I know people who have worked with them, but it's not something that is my area of expertise. So I don't think I could correctly answer that question.
What about you, Jess? Do you have any thoughts?
You're on mute, Jess.
Sorry, I didn't want... Yeah, no, I'd agree really, because I've never worked with them directly either. I know that there's a lot of work going into it and it could be something for the future, but I couldn't really articulate.
Cool. Let's see, do we have any more questions coming through? Not really. I was wondering, Rosie, if you could expand a little bit more about what you were saying with plasmids. So what is a plasmid, and kind of how you mentioned them briefly, but how are they kind of relevant in terms of antibiotic resistance?
So is where it starts to get really complicated. Plasmids are circular pieces of DNA that live independent of a bacterial chromosome, if that makes sense. So you've got the main bit of DNA in a bacteria, and then you've got this circular piece that kind of lives next door, and that can hold like extra genes, and those extra genes can encode for things like antibiotic resistance. They are quite commonly associated with antibiotic resistance, or in my case, they are. And for bacteria, they can quite easily, well, not quite easily, again, it gets really complicated, but they can transfer that plasmid, that circular bit of DNA, from one bacteria to another. And it just makes them able to transfer that information over. That's why antibiotic resistance is so prevalent and why it's such a problem, because it can just move from one bacteria to another.
Interesting. So they can kind of share the genes between themselves.
It's the same way, well, I like to think of it, it's the same way you send a text message to someone, isn't it? Or like a Dropbox. You can share a piece of information, a piece of data with someone, and it can be quite easily picked up. It's the same way in bacteria. Only they have to be in close proximity to each other to be able to do it. They can't just like get on the phone.
Send it over Facebook, it's not an option. So just as we wrap up, could everyone just please remember to do the quiz that Katie posted about what you know, so we can find out what you've learned. And also Scott has put the social media links for Live with Scientists in the chat as well, so everyone please be sure to check those out. And then maybe one final question, Rosie. Who was it that asked this? I think it was Catherine was asking what you think the best way to treat a sort of chronic or recurrent infection is. What's the best strategy for sort of choosing how and what antibiotics to use in a situation like that?
Follow what your doctor says. I think again, there is a set of a set of guidelines for recurrent UTIs. So the NICE guidelines are fantastic for any kind of antibiotic prescribing. So if you did ever want to find out like what is the best way to treat recurrent UTI, or even crazy things like bloodstream infections, the NICE guidelines are the place to go. But I want to say for a recurrent UTI, I feel like coamoxiclav's the first line antibiotic, but I might be wrong about that. It is something that I'd have to check.
Brilliant. Well, it seems like we have no more questions coming through. Should we bring it to a close, Katie?
Yeah, I think that's great. I just wanted to announce that the winner of the quiz was somebody nicknamed Daring Mouse. They know who they are. We've been in contact, and they've actually won a £10 gift voucher for a website called Science Scribbles. So I'll just post a link in the chat there, because even if you didn't win, you can get like really cool stickers on there for like £2 and stuff. So if you are interested, check that out. The link's in the chat. And well done to the person who won. It was a close race. And thank you so much to Jess and Rosie. That was really, really interesting. Just spotlight Jess, and then she can wave you all goodbye as well. Are there any more closing remarks or? No, great. Well, thanks so much for joining us. Check Live with Scientists out on social media, and you can check out Antibiotic Research UK as well. They've got some really interesting information on their website. But yeah, if not, that's bye from us all. Thank you very much.
Thank you, everyone.
Bye.